This is an introduction of my European Commission project that I'm coordinating, and the project was about to develop a point-of-care microfluidic device for the quantification of chemotherapeutic drugs in small body fluid samples by a high selective nanoparticle extraction and liquid crystal detection system. It was, as I said, it's financed by the European Commission in Horizon 2020, and it's a small project. We are, let's say, only few partners, which makes it easy for me to coordinate it, but we have very, very good partners, and we are progressing very well, and it's a pleasure to have this project, to tell the truth. So I will show you a little bit why we started. There is therapeutic drug monitoring, but therapeutic drug monitoring is usually only done in the very beginning during the development of the drugs, but it's very, very difficult, let's say, in clinical practice to provide every patient with proper drug monitoring, because on one hand, the times are very long. On the other hand, usually it needs a specialized laboratory. So there is a lot of different steps, and it needs proper sampling, which is also operator dependent. And the main problem is that most of the therapeutic drugs, especially that one for chemotherapy, are having a very small therapeutic window, meaning that the difference between the active concentration of the drug and the toxic concentration of the drug can be very, very narrow. And usually, let's say, if the patient is not under therapeutic drug monitoring, the problem is that the toxic concentration you only see when you have adverse effects in the patient, and the calculation how chemotherapeutic drugs are calculated are usually either to the body weight or even to the body surface, which is, let's say, quite critical. And let's say, I think I don't have to speak too much about the toxicity of the chemotherapeutic drugs. So talking with medical doctors, we realized that this is one of the unmet clinical needs, that if it's possible to develop, let's say, a small device, very cheap, which allows us to monitor drug concentrations in the clinic without the need to send the samples to another laboratory, especially in very small samples, which is critical for the chemotherapeutic treatment of children with cancer, it will be something which is very welcome in clinical practice, because even the doctors realize that this is something which is needed, but there is no possibility at the moment to have a technique which is able to provide it. So usually the steps are that you have the blood sampling, that, sorry, I need to change my position, I don't see anything. So you have the, thank you. So you have usually the blood sampling by the nurse at the bedside. The sample needs to be, let's say, manufactured very quickly, so it needs to be cooled, it needs to be handled in a proper way. The sampling has to be exactly in the right way, because while the chemotherapeutic drug is still in contact with the blood, there is degradation, there is a change, and this also prevents that there is proper determination of the concentration. So the blood has to be cooled very quickly. There need to be a centrifugation in order to separate the cells, but this has to be handled very carefully, because otherwise a drug which is not free drug but which is inside the cells is usually released because the cells are bursting. And so there are several problems, let's say, in order to do this properly. And as you could see, this is already 2 people involved in this process. And there is already the possibility that the concentration of the drug is changed just during the handling. Then there is this transport to the specialized laboratory. This usually is not causing too many problems. Then the technician in the specialized laboratory is handling it. Most of the techniques are HPLC-MS. Then usually this needs, if it's not an emergency, 1 day. On the next day, usually the results are coming back from the laboratory, and everybody knows that also technicians in laboratories, especially in peak times, with a lot of samples, can do errors. We all think about iron and spinach in this case. And then at the end, on the second day, the medical doctor is making a decision how to, let's say, adjust the concentration of the drug. The main problem could be in this communication pathway, that usually errors can happen, that commas can be shifted and things like this. So there are always problems. First of all, it's a very long way, and only 2 days later the medical doctor is able to adjust treatment. The idea is, with this DiaChemo device, that we have just 1 step. We have the device, we take the sample at the bedside, we get a readout, and the instrument is just communicating, let's say, a value of the actual concentration in the blood to the medical doctor. The idea is to be in the range of 2 hours, not 2 days. As I said, we want a point-of-care device which does this measurement in less than 2 hours in a way that it's easy to handle, so it can be handled by the nurse. It doesn't need a technician. It should be cheap. So let's say for the readout, you have something which which the hospital needs to buy once. The rest can be trashed after usage. And we want to develop a type of platform technology which allows us to adapt the complete system to other drug molecules which can be measured, let's say, with the same technique. So we need, let's say, different modules, and we have, let's say, something which is for the blood sampling, separation, blood cells, plasma. The plasma is then treated with methanol as it is done in the hospital. Then, let's say, we have usually the mixing and with the nanoparticles, or let's say drug treatment, and then the extraction by a liquid phase treatment. And in this liquid phase, usually we want to separate, for example, in case of doxorubicin from doxorubicinol, which is the less active metabolite, but also in other cases, like in case of irinotecan and SN38, we want to be able to distinguish between the prodrug, which is inactive, from the active drug, which is strongly depending on the metabolism of the patient. And so we have a readout. As I said, this is a technology which was developed in collaboration with Stefan Guldin at UCL, with Francesco Stellacci at EPFL, and we filed a patent, and me in Milan, and we filed a patent on this technology using nanoparticles with a functionalized surface, using a technology which was originally developed by Francesco, in which you have a hydrophobic hydrophilic ligand. The ligands are chosen in the way that they are selectively recognizing nanoparticles, creating the chemotherapeutic drugs. They are forming a type of pocket. In this pocket, the drug is trapped, and they are highly specific, so no other drug is entering, no other molecule from the plasma is entering. Once the drug is bound to the nanoparticle surface, we want to use differences in hydrophobicity by separating, let's say, the drug molecules bound to the nanoparticle in a liquid phase, liquid crystal phase separation step. And in this case, we want to read out, let's say, the concentration of the drug in both, in the liquid crystal phase and in the methanol phase, because the idea is that here you see that actually it works. You have a binding, there is a translocation. This is a slightly different mechanism. We are still trying to understand exactly what is happening, but we see that there is a phase separation, and this is specific to doxorubicin in comparison to doxorubicinol, one of the main metabolic compound, and doxorubicinone, which is a second metabolic compound. And as you could see, there is a difference in the separation by these nanoparticles with doxorubicin and doxorubicinol and doxorubicinone. So we find that doxorubicin with the nanoparticles translocate into the liquid crystal phase. The project includes not only the development of the device, but also an evaluation by 2 hospitals. We have a collaboration with a hospital in Krol in Italy for patients, for adult patients, and with the Children's Hospital in Münster in Germany for, especially for this doxorubicin study on babies and small children. And at the end, we hope that we are coming out with a prototype of a device which allows us to separate and measure in very short times, let's say, the chemotherapeutic drug in blood. Thank you for your attention. There's time for one question, only for me. Jesse.
I think it's very, very important study, and if it turns to be successful, which I hope it will be, it will serve many, many purposes. But okay, so you show how you can deal with drugs, but if you have a nanodrugs that actually, when you inject nanodrugs, you have to deal with much more complicated issues because you have the drug and in the free drug, and then you have the drug in the particle itself, and how you will deal with it, with this?
Most probable, this system is only able to measure the free drug.
Absolutely, yeah.
in the blood. Most probable, as you already said for your system, what is really active on the tumor cells is the free drug. So not the doxorubicin, which is still encapsulated in the liposomes, but that one which is free in the tissue or in the blood.
Yeah, if you're doing the tissue, you are absolutely right. You also want to know what's the reservoir, so to get the whole picture, you need both. But I think in the plasma, really, you don't care very much. I mean, you do care for the free drug because of toxicology and less because of efficacy. Because if the drug is released in large quantity in plasma, the drug delivery system has no meaning except of kind of helping to disperse the drug. Otherwise, it has no meaning. So.
No, it's true. I think this system is perhaps extremely interesting to determine the toxic concentration, which is actually that one in plasma if it's getting too high, and it gives you an idea about metabolic activity. On the other hand, I think it's also interesting for a system like irinotecan and SN38 when you have a prodrug which is metabolized into the drug. So in that case, we were already told, also by other medical doctors, that there will be more interesting system in the future. But once we have the platform ready, I think it can be adjusted also also to other drugs. So I think I open now for another 15 minutes, a more general discussion with all the speakers. If you want, join me here. Otherwise, we are running with the microphone. Sorry, too much laboratory. Are there questions? Then I have a question myself to you. Okay. So there's no way, you have to join me on the